Hello everyone, welcome to Bio Academy. I'm Dr. Vasudha. Today we discuss about the possible MCQs from the chapter 10 from the class 11, cell cycle and cell division. These MCQs generated from the NCERT line by line of this chapter. More than 100 MCQs answers with the explanation helps you to better understand of this chapter. Please subscribe our channel Bio Academy to more likely videos. Let us begin with the MCQs. What characteristics defines the starting point of all organism lives? Multicellulatory, single cell origin, external stimuli, environmental adaptation. The answer is single cell origin. Organism begins as single cells, regardless of their eventual size, indicating the fundamental nature of cellular growth in life's beginnings. What question might arise regarding the development of large organism from a single cell? How does a single cell grow? How do cells communicate? How do cells differentiate? How do cells replicate? Answer: how does a single cell grow? The query pertains to the process of scaling up from a single cell to the complex structure of larger organisms, specifically focusing on growth mechanism. What do fundamental traits apply to both cells and all living organisms? Movement and respiration, growth and differentiation, growth and reproduction, homeostasis and adaptation. Answer: growth and reproduction. Cells and living organism shares the characteristic of growth and reproduction, which is essential for their survival and perpetuation. How do cells reproduce? Either by budding, fusion, or division, or differentiation. Answer is by cell division. Cells replicate through division, where each parent cell yields two daughter cells, perpetuating the cellular lineage. What happens after the formation of daughter cells? Apoptosis, senescence, growth and division, differentiation. Answer: growth and division. The offspring cells undergo growth and division, perpetuating the cycle initiated by the parental cell, leading to the formation of a new cell population. Cell division, DNA replication, and cell growth must occur in a coordinated manner during the cell cycle to ensure correct division and formation of progeny cells. What is the term used to describe the sequence of events involving this process? Cellular coordination, genetic replication, cell cycle, progeny formation. Answer is cell cycle. The cell cycle encompasses the sequence of events including DNA replication, cell growth and cell division, ensuring the proper duplication and distribution of genetic material. When does DNA synthesis occur during the cell cycle? During cell division, throughout the entire cycle, or only during cell growth, or during one specific stage? Answer: during one specific stage, the DNA synthesis occurs. DNA synthesis occurs at a specific stage in the cell cycle, ensuring that genetic material is replicated accurately before cell division. How are replicated chromosome DNA distributed to daughter nuclei during cell division? Through random segregation or via cytoplasmic diffusion or by a complex series of events or through direct transport. Answer is by a complex series of events. Replicated chromosomes are distributed to daughter nuclei through a series of intricate events ensuring accurate segregation and formation of genetically identical daughter cells. What controls the events of DNA replication, cell growth and cell division during the cell cycle? Environmental factors, external stimuli, genetic control or cellular metabolism. Answer: genetic control. The events of cell cycle including DNA replication, cell growth and cell division are tightly regulated by genetic mechanism to ensure proper timing and coordination. Which process is a continuous aspect of cell growth during the cell cycle? Cell replication, cell division, cytoplasmic increase or chromosome distribution? Answer: cytoplasmic increase. While DNA replication occurs at a specific stage, cell growth in terms of cytoplasmic increase is continuous throughout the cell cycle. What is the approximate duration of the cell cycle for human cells in culture? 12 hours, 24 hours, 48 hours or 72 hours? Answer: 24 hours. Human cells in culture typically undergo one cell division approximately every 24 hours. How long does it take for each cell to propagate or progress through the cell cycle? About 30 minutes, about 60 minutes or about 90 minutes or about 120 minutes? Answer: about 90 minutes. Each cells can progress through the cell cycle in approximately 90 minutes, indicating a faster cell cycle compared to human cell. What are the two basic phases of the cell cycle? G1 phase and G2 phase, S phase and G2 phase, interphase and mitosis phase, or prophase and telophase? Answer is interphase and mitosis phase. The cell cycle consists of interphase and M phase or mitosis phase. During which phase of the cell cycle does actual cell division or mitosis occur? G1 phase, S phase, G2 phase or M phase? Answer is M phase. Mitosis occurs during the M phase of the cell cycle. What percentage of the cell cycle duration does interphase typically occupy in human cell? Less than 10%, approximately 25%, more than 50%, more than 95%. Answer is more than 95%. Interphase occupies more than 95% of the duration of the cell cycle in human cells, with cell division proper mitosis lasting for only about 1 hour. Majority of the cell cycle is the interphase. What characteristic the beginning of the M phase in the cell cycle? Cytoplasmic division, DNA replication, nuclear division or cell growth? Answer is nuclear division. The M phase starts with the separation of daughter chromosomes or karyokinesis, which corresponds with the nuclear division. What process usually marks the end of the M phase in the cell cycle? DNA replication, cytoplasmic division, cell growth, nuclear division. Answer is cytoplasmic division. The M phase usually ends with the division of cytoplasm, also known as cytokinesis, completing the process of the cell division. What characterizes the interface of the cell cycle? Resting phase with no activity, continuous cell growth and DNA replication, nuclear division and cytoplasmic division, DNA synthesis. The answer is continuous cell growth and DNA replication. In the interface, what happened? Here, the interface is a period of preparation of the cell division involving both cell growth and DNA replication. How many phases does the interface of the cell cycle consist of? 1, 2, 3 or 4? The answer is 3. The interface is divided into 3 phases, that are G1 phase or gap 1, S phase or synthesis phase, and G2 phase or gap 2 phase. Which phase of interface involves DNA replication? G1 phase, S phase, G2 phase or M phase? Answer is S phase. The S phase or synthesis phase is the period during which DNA replication occurs or DNA synthesis process is occurred in preparation of cell division. What characterizes the G1 phase of the cell cycle? DNA replication, metabolic inactivity, continuous cell growth, initiation of mitosis. The answer is metabolic inactivity. G1 phase is a period of metabolic activity and continuous cell growth but does not involve DNA replication. What occurs during the S phase of the cell cycle? Nuclear division, DNA replication or protein synthesis or centriole duplication. Answer is DNA replication. The S phase is when DNA synthesis or replication takes place, resulting in the doubling of DNA content in the cell. How does the DNA content change during the S phase? It halves, it quadruples, or it remains the same, or it replicates. Answer is it quadruples. That means the amount of DNA per cells doubles during the S phase from 2C to 4C, 2 set to 4 set. What happens in animal cell during the S phase? Protein synthesis, centriole duplication, initiation of mitosis, nuclear division. The answer is centriole duplication. In animal cell, DNA replication begins in the nucleus and the centrioles duplicate in the cytoplasm during the S phase. What characterizes the G2 phase of the cell cycle? DNA replication, nuclear division, protein synthesis or cell growth and protein synthesis. Answer is cell growth and protein synthesis. In G2 phase is a period of cell growth and protein synthesis in preparation for mitosis, following DNA replication in the S phase. In which stage of the cell cycle do cells enter when they cease dividing and become metabolically active but no longer proliferate unless called upon to do so? G1 phase, G2 phase, S phase, G0 phase. Answer is G0 phase. Cells that ceases dividing and become metabolically active but do not proliferate enter the G0 phase, also known as the quiescent stage. In animals, where is mitotic cell division primarily observed? Germ cells, somatic cells, muscle cells, nerve cells. Answer is somatic cell. Mitotic cell division is primarily observed in somatic cells of animals. Which organism exhibit mitotic division in haploid cells, contrary to the general pattern seen in animals? Male honeybee, female honeybees, male fruit flies, female fruit flies. The answer is male honeybees. In male honeybees, also known as drones, exhibit mitotic divisions in haploid cells, which is an exception to the general pattern observed in animals. In plants, where can mitotic division be observed in both haploid and diploid cells? Embryo, seed, gametophyte, sporophyte. Answer is gametophyte. Mitotic divisions in both haploid and diploid cells can be observed during the gametophyte stage in plants as part of the alteration of generation life cycle. During which phase of the cell cycle do cells primarily undergo growth and preparation for DNA replication? G1 phase, S phase, G2 phase, M phase. Answer is G1 phase. G1 phase is primarily dedicated to cell growth and preparation for DNA replication in the cell cycle. Which phase of the cell cycle is described as the most dramatic period involving a major recognition of virtually all components of the cell? G1 phase, S phase, G2 phase or mitosis phase? Answer is mitosis phase. Mitosis is described as the most dramatic period of the cell cycle involving a major reorganization of virtually all components of the cell. What is another term for mitotic cell division? Considering the number of chromosomes in parent and progeny cell remains the same. Equational division, unequal division, meiotic division or diploid division? Answer is equational division. Mitotic cell division, where the number of chromosomes in parents and progeny cells remains the same, is also referred as the equational division. How many stages are typically recognized during karyokinesis or nuclear division in mitosis? 2, 3, 4 or 5? The answer is 4. Karyokinesis or nuclear division in mitosis is typically divided into 4 stages, that is prophase, metaphase, anaphase and the telophase. What characterizes the process of karyokinesis during mitosis? Progressive and discontinuous, progressive and continuous or regressive and discontinuous, regressive and continuous? The answer is progressive and continuous. Karyokinesis during mitosis is described as a progressive process and clear-cut lines cannot be drawn between various stages. Which stage of mitosis involves the condensation of chromosomes and the disappearance of the nuclear membrane? Prophase, metaphase, anaphase, telophase? The answer is prophase. Prophase is a stage of mitosis where chromosome condenses, becoming visible, and the nuclear membrane begins to disappear. Which phase of the cell cycle proceeds prophase during mitotic karyokinesis? The answer is G2 phase. Prophase follows the S and G2 phase of interphase in cell cycle. What is the primary characteristic event marking the initiation of prophase during mitosis? Duplication of centromeres, formation of mitotic chromosomes, disappearance of the nuclear membrane, initiation of DNA replication. The answer is formation of mitotic chromosomes. Prophase follows the S and G2 phase of interphase in the cell cycle. What structural features distinguishes mitotic chromosome during prophase? Single chromatid structure, double chromatid structure, absence of centromeres, presence of telomeres. Answer is double chromatid structure. Mitotic chromosomes during prophase are composed of two chromatids attached together at the centromere. What cellular structure undergoes duplication during the S phase of interphase and begins to move towards opposite poles of the cells during prophase? Golgi apparatus, endoplasmic reticulum, centrosomes or lysosome? The answer is centrosomes. The centrosomes undergo duplication during the S phase of interphase and begins to move towards opposite poles of the cell during prophase. What microtubule structure radiate out from the centrosome during prophase, contributing to the formation of the mitotic apparatus? A flagella, cilia, aster or spindle fibers? Answer is asters. Asters are microtubule structures that radiate out from the centrosomes during prophase, contributing to the formation of mitotic apparatus along with spindle fibers. Which cellular structures are absent in cells at the end of prophase when viewed under the microscope? Golgi complexes, endoplasmic reticulum, nucleus or nuclear envelope? Answer is all the four options are correct. Cells at the end of prophase lack Golgi complexes, endoplasmic reticulum, nucleus and the nuclear envelope. And these structures are disassembled during prophase in the preparation for mitotic division. Which phase of the cell cycle directly follows prophase? Metaphase, anaphase, telophase or interphase? The answer is metaphase. Metaphase directly follows the prophase in the sequence of mitotic phases. What is the primary event that occurs during prophase in mitotic karyokinesis? Separation of sister chromatids, alignment of chromosome at the cell equator, condensation of chromosomal material, disassembly of the mitotic spindle. The answer is condensation of chromosomal material. The primary event in prophase is the condensation of chromosomal material to form compact mitotic chromosomes. What cellular component is responsible for organizing the mitotic spindle during prophase? Centrosomes, Golgi apparatus, endoplasmic reticulum or nucleus? The answer is centrosome. The centrosome plays a key role in organizing the mitotic spindle during prophase. What structural feature distinguishes mitotic chromosomes during prophase? Single chromatid structure, double chromatid structure, absence of centromere, presence of telomeres. The answer is double chromatid structure. Mitotic chromosome during prophase are composed of two chromatids attached together at the centromeres. What marks the beginning of the second phase of mitosis? Complete disintegration of the nuclear envelope, condensation of chromosome, formation of the mitotic spindle, duplication of centrosomes. The answer is complete disintegration of the nuclear envelope. The complete disintegration of the nuclear envelope marks the beginning of the second phase of the mitosis. What characterizes the morphology of chromosomes during metaphase? Condensation, dispersion, alignment at the equator, duplication. The answer is alignment at the equator. During metaphase, chromosomes align at the equator of the cell. What characterizes the morphology of chromosome during metaphase? Condensation, dispersion, alignment at the equator, or duplication? The answer is alignment at the equator. During metaphase, chromosomes align at the equator of the cell. What structures serve as a site of attachment of spindle fiber to chromosome during metaphase? Centromeres, telomeres, nuclei, or kinetochore? The answer is kinetochore. Kinetochore structure serves as the site of attachment of spindle fibers to chromosome during metaphase. What is the plane of alignment of chromosome during metaphase referred to as prophase plate, metaphase plate, anaphase plate, or telophase plate? The answer is metaphase plate. The plane of alignment of chromosome during metaphase is referred to as the metaphase plate. What characterizes the attachment of sister chromatids to spindle fibers during metaphase? Both chromatids attached to fibers from the same pole. One chromatid attached to fiber from one pole, the other attached to fiber from the opposite pole. Both chromatids attached to fibers from opposite poles. Chromatids not attached to the spindle fibers. The answer is one chromatid attached to the fibers from one pole and other attached to the fibers from the opposite pole. In metaphase, one chromatid of the each chromosome is connected by its kinetochore to spindle fibers from one pole, while its sister chromatid is connected by its kinetochore to spindle fibers from the opposite pole. What structures do spindle fibers attach to during mitosis? Centromeres, nucleoli, kinetochore, or Golgi apparatus? The answer is kinetochore. Spindle fibers attached to the kinetochore of chromosome during mitosis. How do chromosomes get aligned along the metaphase plate during metaphase? Through condensation, through centriole movement, through spindle fiber attachment, or through nuclear envelope disintegration? The answer is through spindle fiber attachment. Chromosomes get aligned along the metaphase plate during metaphase through the attachment of spindle fibers. What is the function of spindle fibers during mitosis? chromosome condensation, nuclear envelope disintegration, chromosome alignment and segregation, centriole duplication. The answer is chromosome alignment and segregation. Spindle fibers play a crucial role in aligning and segregating chromosome during mitosis. Where do chromosomes align during metaphase? Prophase plate, metaphase plate, anaphase plate, telophase plate. The answer is metaphase plate. Chromosome align along the metaphase plate during metaphase. Which cellular event characterizes the beginning of metaphase plate? Complete disintegration of the nuclear envelope, duplication of centrosomes, condensation of chromosomal material, alignment of chromosome at the equator. The answer is alignment of chromosomes at the equator. The alignment of chromosome at the equator marks the beginning of metaphase in mitosis. What characterizes the onset of anaphase in mitosis? Alignment of chromosome at the equator, condensation of chromosomal material, separation of sister chromatids, disintegration of nuclear envelope. The answer is separation of sister chromatids. The onset of anaphase is characterized by the separation of sister chromatids. What term is used to describe the two daughter chromatids resulted from the separation of sister chromatids during anaphase? Chromosomes, centrosomes, kinetochore, daughter chromosomes. The answer is daughter chromosomes. The two daughter chromatids resulting from the separation of sister chromatids are referred to as the daughter chromosome during anaphase. How do daughter chromatids move during anaphase? Towards each other, towards the equator, towards the centromere, towards opposite pole. The answer is towards opposite pole. Daughter chromatids move towards opposite pole during anaphase. What happens to the centromeres during anaphase? They duplicate, they disappear, they split, they condense. The answer is they split. Centromeres split during anaphase, allowing sister chromatids to separate. How are the arms of chromosome positioned relative to the centromeres during anaphase? Leading the centromeres, trailing behind the centromeres, attached to the centromere or surrounding the centromeres. The answer is trailing behind the centromeres. During anaphase, the arms of the chromosome trail behind the centromeres as the chromatids move towards opposite poles. How are the arms of chromosome positioned relative to the centromeres during anaphase? Condensation of chromosomal material, disintegration of the nuclear envelope, decondensation of chromosome, alignment of chromosome at the equator. The answer is decondensation of chromosomes. Telophase begins with the decondensation of chromosome as they reach their respective poles. What happens to the individual chromosomes during telophase? They condense further, they lose their individuality, they replicate, they align at the equator. The answer is they lose their individuality. During telophase, individual chromosomes lose their individuality as discrete elements. What structures reform during telophase? Centromeres, kinetochore, nuclear envelope, spindle fiber. The answer is nuclear envelope. During telophase, the nuclear envelope develops around the chromosome clusters at each pole, forming two daughter nuclei. What cellular components reform during telophase, indicating the end of mitosis? Nucleolus, Golgi complex, and endoplasmic reticulum, or centrioles and spindle fiber, centromeres and kinetochore, or mitotic spindle. The answer is nucleolus, Golgi complex, and endoplasmic reticulum. The nucleolus, Golgi complex, and endoplasmic reticulum reform during telophase, indicating the end of mitosis. What term is used to describe the formation of two daughter nuclei during telophase? Centromere duplication, karyokinesis, cytokinesis, nucleokinesis. The answer is karyokinesis. The formation of two daughter nuclei during telophase is referred to as karyokinesis. What term describes the process of dividing the cytoplasm at the end of mitosis? Karyokinesis, cytokinesis, nucleokinesis, telophase. The answer is cytokinesis. Cytokinesis is the process of dividing the cytoplasm at the end of mitosis. How is cytokinesis achieved in animal cell? Formation of cell plate, formation of cleavage furrow, formation of a cell wall, separation of the centromeres. The answer is formation of a cleavage furrow. In animal cells, cytokinesis is achieved by the appearance of a furrow in the plasma membrane, which gradually deepens and ultimately divides the cell cytoplasm into two. How do plant cells undergo cytokinesis? Formation of cleavage furrow, formation of cell plate, formation of a cell wall, formation of a cell membrane. The answer is formation of cell plate. In plant cells, cytokinesis occurs through the formation of cell plate, which starts in the center of the cell and grows outwards to meet the existing lateral walls. What is the precursor to the new cell wall formed during cytokinesis in plant cell? Middle lamella, plasma membrane, cell membrane, Golgi complex. The answer is middle lamella. The precursor to the new cell wall formed during cytokinesis in plant cell is called as the middle lamella. In some organism, what condition arises when karyokinesis is not followed by cytokinesis? Cictium, cleavage furrow, cell plate, centriole duplication. The answer is Cictium. When karyokinesis is not followed by the cytokinesis leading to the formation of a multinucleated condition, it is termed as Cictium. Cictium. In which organisms beside diploid cells does mitosis also occur? Higher plants only, social insect only, both lower plants and social insects only in diploid cells. The answer is both lower plants and social insects. Mitosis occurs not only in diploid cell but also in haploid cells of lower plants and social insects. What is the significance of mitosis in the growth of multicellular organism? It restores the nucleocytoplasmic ratio. It repairs the damaged cells. It produces haploid daughter cells. It results in diploid daughter cells. The answer is it results in diploid daughter cells. Mitosis leading to the production of diploid daughter cells contributing to the growth of multicellular organisms. Which cell in the human body are constantly replaced through mitotic division? Liver cell, brain cell, blood cell, muscle cell. The answer is blood cells. Mitotic division constantly replaces the blood cells in the human body. What is the role of mitotic division in meristematic tissue of plants? Repair damaged tissues, produce haploid spores, stimulate growth throughout the plant's life, form reproductive structure. The answer is stimulate growth throughout the plant's life. Mitotic division in meristematic tissue such as apical and lateral cambium contribute to continuous growth throughout the plant's life. Which statement best describes the genetic complement of daughter cells produced by mitosis? Haploid daughter cells with different genetic composition, haploid daughter cell with identical genetic composition, diploid daughter cell with different genetic composition, diploid daughter cells with identical genetic composition. The answer is diploid daughter cells with identical genetic composition. Mitosis typically results in the production of diploid daughter cells with identical genetic composition to the parent cell. Which statement best describes the genetic complement of daughter cells produced by mitosis? Mitosis, fertilization, meiosis, gametogenesis. The answer is meiosis. Meiosis is the process that ensures the production of haploid daughter cells in sexually reproducing organism. How many cycles of DNA replication occur during meiosis? 1, 2, 3 or 4? The answer is 1. Meiosis involves only a single cycle of DNA replication. What event initiates meiosis 1? DNA replication, pairing of homologous chromosome, recombination between non-sister chromatids, formation of haploid cell. The answer is pairing of homologous chromosomes. Meiosis 1 is initiated after the parental chromosomes have replicated to produce identical sister chromatids, and it involves the pairing of homologous chromosomes. How many haploid cells are formed at the end of meiosis second? 1, 2, 3, 4. The answer is 4. 4 haploid cells are formed at the end of meiosis second. Which phase of meiosis involves the separation of homologous chromosomes and sister chromatids respectively? Prophase 1 and prophase 2, metaphase 1 and metaphase 2, anaphase 1 and anaphase 2, telophase 1 and telophase 2. The answer is anaphase 1 and anaphase 2. Anaphase 1 involves the separation of homologous chromosome, while anaphase 2 involves the separation of sister chromatids. What is the process called when chromosomes start pairing together during prophase 1? Synthesis, synapsis, synthesis complex, synaptonemal complex. The answer is synapsis. The process of chromosome pairing together during prophase 1 is called as synapsis. What is the complex structure formed during synapsis? Synthesis complex, synaptonemal complex, chromosomal complex, tetrad complex, The answer is synaptonemal complex. Synaptonemal complex is the complex structure formed during synapses. What are paired chromosome during synapses? Homologous chromosome, sister chromatids, tetrad chromosome, or recombinant chromosome? The answer is homologous chromosomes. Paired chromosomes during synapses are called as homologous chromosomes. When do recombination nodules appear during prophase 1? Leptotin, zygotin, pachytin, diplotin. The answer is pachytin. Recombination nodules appear during the pachytin stage of prophase 1. What enzyme is involved in the process of crossing over during prophase 1? Recombinant, synthetase, recombinase, synapse. The answer is recombinase. Crossing over during prophase 1 is an enzyme mediated process and the enzyme involved is called recombinase. Next. What marks the beginning of diplotin during mitotic prophase 1? Dissolution of the nuclear envelope, dissolution of the synaptonemal complex, condensation of chromosome, formation of chiasmata. The answer is dissolution of synaptonemal complex. The beginning of diplotin is recognized by the dissolution of the synaptonemal complex. What structures are formed at the site of crossover between homologous chromosome during diplotin? Homologous chromosome, sister chromatid, chiasmata, synaptonemal complexes. The answer is chiasmata. The X-shaped structure called as chiasmata are formed at the site of crossover between homologous chromosome during diplotin. In some vertebrates, how long can diplotin last in oocysts? Hours, days, months or year? Diplotin does not occur in oocysts. The answer is months or year. In some vertebrates, diplotin can last for months or year in oocytes. What marks this transition from diakinesis to metaphase during meiotic prophase 1? Dissolution of the nuclear envelope, formation of chiasmata, terminalization of chiasmata, formation of synaptonemal complex. The answer is terminalization of chiasmata. The final stage of meiotic prophase 1 diakinesis is marked by the terminalization of chiasmata, which represents the transition to metaphase. What event characterizes metaphase 1 of meiosis? Dissolution of the nuclear envelope, alignment of bivalent chromosome on equatorial plate, formation of chiasmata, formation of synaptonemal complex. The answer is alignment of bivalent chromosome on equatorial plate. Metaphase 1 is characterized by the alignment of bivalent chromosome on the equatorial plate where microtubules from opposite spindle poles attach to the kinetochores of homologous chromosome. What event characterizes metaphase 1 of meiosis? Dissolution of the nuclear envelope, alignment of bivalent chromosomes on equatorial plate, formation of chiasmata, formation of synaptonemal complex. The answer is alignment of bivalent chromosome on equatorial plate. Metaphase 1 is characterized by the alignment of bivalent chromosome on equatorial plate where microtubules from opposite spindle poles attached to the kinetochores of homologous chromosome. What occurs during anaphase 1 of meiosis? Sister chromatid separates, homologous chromosomes separate, nuclear membrane reappears, nucleolus reappears. The answer is homologous chromosomes separate. During anaphase 1 of meiosis, homologous chromosomes separate and move towards opposite poles of the cell. What follows telophase 1 of chromosome? Interphase, prophase second, anaphase second, metaphase second. The answer is prophase second. Prophase second follows the telophase 1 of meiosis. What term is used to describe the pair of cells formed after telophase 1 of meiosis? Monads, dyads, triads or tetrads? The answer is dyads. The pair of cells formed after telophase 1 of the meiosis is called as dyads. What characterizes the stage between the two meiotic division? Synapsis, interphase, anaphase or metaphase? The answer is interphase. The stage between two meiotic division is called as the interphase. What follows interkinesis in the meiotic prophase? Prophase 1, prophase 2, metaphase 1, metaphase 2. The answer is prophase 2. Prophase 2 follows interkinesis in the meiotic process. When does meiosis second typically begin? After prophase second, after metaphase second, after anaphase 1, before and the chromosomes fully elongate. The answer is before the chromosome fully elongate. Meiosis second is initiated immediately after cytokinesis, usually before the chromosome have fully elongated. What happens to the nuclear membrane by the end of prophase second? It reappears, it elongates, it disappears, it becomes fragmented. The answer is it disappears. By the end of prophase second, the nuclear membrane disappears. What characterizes metaphase second of meiosis? Alignment of homologous chromosome at the equator, alignment of sister chromatids at the equator, separation of homologous chromosome, separation of sister chromatids. The answer is alignment of sister chromatids at the equator. Metaphase second is characterized by the alignment of sister chromatids at the equator. What initiates anaphase second of meiosis? Dissolution of the nuclear membrane, splitting of centromere, alignment of chromosome at the equator, attachment of microtubules to the kinetochore. The answer is splitting of the centromere. Anaphase second begins with the simultaneous splitting of the centromere of each chromosome. What facilitates the movement of sister chromatid towards opposite pole during anaphase second? Attachment of microtubules to the kinetochores, formation of the nuclear membrane, synapsis of homologous chromosome, formation of the synaptonemal complex. The answer is attachment of microtubules to the kinetochores. The movement of sister chromatids towards opposite pole during anaphase second is facilitated by the shortening of microtubules attached to the kinetochores. What stage marks the end of meiosis? Prophase second, metaphase second, anaphase second, or telophase second? The answer is telophase second. Meiosis ends with telophase second. What occurs during telophase second of meiosis? Chromosome alignment at the equator, chromosome separate and move towards opposite poles, nuclear envelope reappears, synaptonormal complex forms. The answer is nuclear envelope reappears. During telophase second, the two groups of chromosome get enclosed by a nuclear envelope. What follows telophase second in the meiotic process? Interphase, prophase 1, cytokinesis, anaphase second. Cytokinesis. Cytokinesis follows telophase second in the mitotic process. How many daughter cells are formed at the end of meiosis? 2, 3, 4, 5. The answer is 4. Meiosis results in the formation of 4 haploid daughter cells. What is the term used to describe the group of 4 daughter cells formed at the end of meiosis? Monads, dyads, triads, tetrads. The answer is tetrads. The formation of 4 haploid daughter cells is referred to as a tetrad. What is the main purpose of meiosis in sexually reproducing organism? To increase chromosome number, to decrease genetic variability, to achieve conservation of specific chromosome number, to eliminate genetic variability. The answer is to achieve conservation of specific chromosome number. Meiosis ensures the conservation of specific chromosome number in sexually reproducing organism. What is the paradoxical outcome of meiosis? Increase in chromosome number, decrease in chromosome number, decrease in genetic variability, increase in genetic variability. The answer is decrease in chromosome number. Meiosis results in the reduction of chromosome number by half, which is a paradoxical outcome consisting its role in reproduction. How does meiosis contribute to genetic variability in population? By reducing chromosome number, by increasing chromosome number, by eliminating genetic variations, by promoting recombination and crossing over. The answer is by promoting recombination and crossing over. Meiosis increases genetic variability through processes such as recombination and crossing over. Why are variations important for the process of evolution? Variations decrease adaptability. Variation increases chromosome number. Variation decreases genetic variability. Variation increases adaptability to changing environments. The answer is variation increases adaptability to change environments. Meiosis increases genetic variability through processes such as recombination and crossing over. Why are variations important for the process of evolution? Variations decreases adaptability. Variation increases chromosome number. Variation decreases genetic variability. Variation increases adaptability to changing environments. The answer is variations increases adaptability to changing environments. Variation provide organisms with different traits, increasing their adaptability to changing environments, which is crucial for the process of evolution. What is the result of cytokinesis following telophase second of meiosis? formation of diploid daughter cells, formation of tetrad of cells, formation of triploid daughter cells, formation of haploid daughter cells. The answer is formation of tetrad of cells. Cytokinesis following telophase second of meiosis result in the formation of a tetrad of cell, each being haploid. What is the result of cytokinesis following telophase second of meiosis? Formation of diploid daughter cell, formation of tetrad of cell, formation of triploid daughter cells, formation of haploid daughter cells. The answer is formation of tetrad of cells. Cytokinesis following telophase second of meiosis result in the formation of the tetrad of cell, each being
Thank you for
haploid.
watching. Let's meet in another video. Till then, take care. Bye bye.